Good day everyone, my name is Rian B. Bactong and my topic is all about cell structure, chapter 3. And without further ado, let us know what is cell. So, cell in biology is the basic structural unit of life. It is the smallest unit capable of independent existence which can reproduce itself exactly. So, all living organisms, which the exception of uh, viruses, are composed of one or more cells. So, single-cell organisms such as bacteria, protozoa, and other microorganisms are termed unicellular, and while plants and animals, which contain many cells, are termed multicellular organisms. So, highly complex organisms, such as human beings, consist of billions of cells and are organized into tissues and organs. So, although these cells may differ widely in size, appearance, and function, their essential features are similar. So, cell divide by mitosis or by meiosis. So, when gametes are being formed, the cytoplasm of all cells contain ribosomes, which carry out protein synthesis that we'll tackle later, and DNA, the coded instructions of for the behavior and reproduction of the cell, and the chemical machinery for the translation of these instructions into the manufacture of proteins. So viruses lack this translation machinery, also have parasiticize cells in order to reproduce themselves. So it is important to learn about the cell theory because we're talking about the discovery of the cell, but it also represented a major evolution in microscopes that allowed scientists to visualize what they were hypothesizing in laboratory. So now that they can actually see cells, they could prove that they existed. Now these cells help define the characteristic of life. So, cell theory actually dates back to the 17th century with Robert Hooke. He was a man who worked for the Royal Society in England, and he was a very prominent member of the Royal Society. And Robert Hooke published Micrographia, a book filled with drawings and descriptions of the organisms he viewed under the recently invented microscope. So he was a philosopher, but he also did science experiments, and at that time no one knew about cells. So he was actually the first one to discover cells, and he did so using very old-fashioned microscope. So the invention of the microscope led to the discovery of the cell by Hooke. And while looking at cork, Hooke observed back-shaped structures which he called cells, as they reminded him of the cells or rooms in monasteries. So this discovery led to the development of the classical cell theory. Shortly after Hooke published his result, a man by the name of Anton van Leeuwenhoek, he was much more skilled at building microscopes, and he was actually able to build one with 300 times the magnification, which impressed everyone. He took also protozoa and bacteria, which he didn't know were protozoa and bacteria at that time, but he named them animalcules. He reported to the Royal Society in the 1600s that because these these were model organisms and that represented a quality of life, that these specimens were living organisms. He is also credited for making the first observations of human blood cells and, very importantly, sperm cells, because he was able to observe that sperm and egg were fusing together to create a new generation of cells. It is important because it was the evidence against spontaneous generation, that cells were not just arising out of nowhere, that they were coming from pre-existing cells. The work of Hooke and Leeuwenhoek, who were the ones who really solidified that cells existed. The cell theory didn't really come into existence, the theory itself, until the mid 1800s. So scientists at that time were recognizing that cells existed, but the theory about these cells represented and what they stood for in uh, forms of life was just beginning to surface. Finally, in the mid 1800s, these three German men, Matthias Schelden, Theodor Schwann, and Rudolf Virchow. So, cell theory, a fundamental scientific theory of biology, according to which cells are held to be the basic units of all living tissues, first proposed by German scientists Theodor Schwann and Matthias Schelden. The theory that all plants and animals are made up of cells marked a great conceptual advance in biology and resulted in renewed attention to the living process that go on in cells. When Matthias Jacob Sheldon, interested in plant anatomy, stated that the lower plants all consist of one cell, while the higher ones are composed of many individual cells. When Theodor Schwann, Sheldon's friend, extended the cellular theory to include animals, he thereby brought about a rapprochement between botany and geology. The two scientists clearly stated in that cells are the elementary particles of organisms in both plants and animals, and recognize that some organisms are unicellular and other multicellular. So these Sheldon's contributions on plants were acknowledged by Schwann as the basis for his comparison of animal and plant structure. While Rudolf Virchow stated in German omnis cellula il cellula, which means cells arise from already existing cells. These three men were finally able to come together with a large enough body of work and put together a concept known as the cell theory, which is actually a group of concepts. So the three cell theories are: all living organisms are composed of one or more cells. Cell is the basic structural and functional unit of living organisms, and all cells arise from pre-existing cells. Now let us proceed to molecular structure of a cell membrane. Cell membranes protect and organize cells. All cells have an outer plasma membrane that regulates not only what enters the cell, but also how much of any given substance comes in. Unlike prokaryotes, eukaryotic cells also possess internal membranes that encase their organelles and control the exchange of essential cell components. Both types of membranes have a specialized structure that facilitates their gatekeeping function. Cell membrane serves as barriers and gatekeepers. They are semi-permeable, which means that some molecules can diffuse across the lipid bilayer but others cannot. Small hydrophobic molecules and gases like oxygen and carbon dioxide cross membranes rapidly. Small polar molecules, such as water and ethanol, can also pass through membranes, but they do more slowly. On the other hand, cell membranes restrict diffusion of highly charged molecules, such as ions, and large molecules, such as sugars and amino acids. The passage of these molecules relies on specific transport proteins embedded in the membrane. One key feature of a cell is the cell membrane or plasma membrane, which separates what's inside the cell from what's outside of the cell. This is a very important function because without this separation, there could be no such thing as life, because an organism must be distinct from its surroundings. Inside a cell is where metabolism takes place and genetic information is stored, so under regulated conditions or homeostasis. Most of membrane is made of molecules called phospholipids. It have a head made of phosphate and two tails made of fatty acids. The heads are hydrophilic, meaning that they are attracted to water. The tails are hydrophobic, meaning that they repel water. Hydrophobic means water-fearing. So because of these properties, phospholipids from two layers known as phospholipid bilayer with hydrophobic tails facing each other, and the hydrophilic heads facing watery external environment and the watery cytoplasm inside of the cell. A second membrane is cholesterol. Although people often think of cholesterol as a bad thing, it's actually a very important part of cell membranes, where it helps to stabilize the membrane and make it better at controlling what can pass through it. So proteins are another important part of the membrane. They do many functions, but one of the most important is transporting materials across the membrane. Especially, they have a hard time passing through the phospholipid bilayer. A fourth membrane component is carbohydrates. These are often involved in cell identification. Examples are blood types are the result of membrane carbohydrates attached to proteins. These help you to identify blood that belongs to you and distinguish it from foreign invaders. So, all of these components work together to make the plasma membrane that surrounds all cells a critically important barrier needed for all life on Earth. So, it is a selective permeable membrane which allows certain materials to pass through it, allows essential to enter to the cell, such as water and oxygen. Also, cell membrane remove toxic substance out of the cell like carbon dioxide and waste products. Cell membrane allow good products to enter to the cell and remove waste products out of the cell. Now let us proceed to structural and function of cellular organelles. First are nucleus. So nucleus is the central membrane enclosed part of a eukaryotic cell containing threads of DNA. During cell division, these coil up to form chromosomes. The nucleus controls the function of the cell by determining which proteins are produced within it. So because proteins are the chief structural molecules of living matter, and as enzymes regulate all aspects of metabolism, it may be seen that the genetic code within the nucleus is effectively responsible for building and controlling the whole organism. So the nucleus contains the nucleolus, the part of the cell where ribosomes are produced. So movement of molecules into and out of the nucleus occurs through the nuclear pores. So an average mammalian nucleus has approximately 3,000 pores. Next are cell membrane or plasma membrane, a thin layer of protein and fat surrounding cells. It controls substances passing between the cytoplasm and the intercellular space. The cell membrane is semi-permeable, allowing some substance to pass through and some not. So generally, small molecules such as water, glucose, and amino acids can penetrate the membrane, while large molecules such as starch cannot. So membranes also play a part in active transport, hormonal response, and cell metabolism. Next are ribosome. So ribosome in biology is the protein-making machinery of the cell. Ribosomes are located in the endoplasmic reticulum or ER of eukaryotic cells and are made of proteins and a special type of RNA. Ribosome RNA, they receive messenger RNA copied from the DNA and amino acids and translate the messenger RNA by using its chemically coded instructions to link amino acids in a specific order to make a strand of a particular protein. Next are cytoplasm, the part of the cell outside the nucleus. Strictly speaking, this includes all the organelles or the mitochondria or the chloroplast and so on. but often cytoplasm refers to the jelly-like matter in which the organelles are embedded, correctly termed the cytosol. The cytoplasm is the site of protein synthesis. In many cells, the cytoplasm is made up of two parts: the ectoplasm or plasma gel, a dense gelatinous outer layer concerned with cell movement, and the endoplasm or plasmasol, a more fluid inner part where most of the organelles are found. Next are vacuole. In biology, a fluid-filled membrane-bound cavity inside a cell. It may be a reservoir for fluids that the cell will secrete to the outside, or may be filled with excretory products or essential nutrients that the cell needs to store. So, plant cells usually have a large central vacuole containing sap or sugar and salts in solution, which serves both as a store of food and as a key factor in maintaining rigor in amoeba. Single-celled animals or vacuoles are the sites of digestion or engulfed food particles. Lysosome. Lysosome is a membrane-enclosed structure or organelle inside a cell, principally found in animal cells. Lysosomes contain enzymes that can break down proteins and other biological substances. They play a part in digestion and in the white blood cells known as phagocytes. The lysosome enzymes attack indigested bacteria. Next are mitochondria. So, mitochondria are singular mitochondrion, membrane-enclosed organelles within eukaryotic cells, containing enzymes responsible for energy production during aerobic respiration. And mitochondria absorb and glucose and produce energy in the form of ATP by breaking down the glucose. So these rod-like or spherical bodies are thought to be derived from free-living bacteria that at a very early stage in the history of life invaded larger cells and took up a symbiotic way of life inside. Each cell contains its own small loop of DNA called mitochondrial DNA, and new mitochondria arise by division of existing ones. Mitochondria each have 37 genes. So an operon is a group of prokaryotic genes with a related function that are often grouped and transcribed together. In addition, the operon has only one promoter region for the entire operon. So the DNA of prokaryotes is organized into a circular chromosome, supercoiled within the nucleus region of the cell's cytoplasm. Proteins that are needed for the specific function or that are involved in the same biochemical pathway are encoded together in blocks called operons. For example, all of the genes needed to use lactose as an energy source are coded next to each other in the lactose or lac operon and transcribed into a single mRNA. So, repressor and activators are proteins produced in the cell. Both repressor and activators regulate gene expression by binding to specific DNA sites adjacent to the genes they control. In general, activators bend to the promoter site while repressor prevent transcription of a gene in response to an external stimulus, whereas activators increase the transcription of a gene in response to an external stimulus. Endorsers are small molecules that may be produced by a cell or that are in the cell's environment. Endorsers either activate or repress transcription depending on the needs of the cell and the availability of substrate. So bacteria such as Escherichia coli need amino acids to survive and are able to synthesize many of them. Tryptophan is one such amino acid that E. coli can either ingest from the environment or synthesize using enzymes that are encoded by 5 genes. These 5 genes are next to each other in what is called the tryptophan operon. The genes are transcribed into a single mRNA, which is then translated to produce all 5 enzymes. If tryptophan is present in the environment, then E. coli does not need to synthesize it and the operon is switched off. However, when tryptophan availability is low, the switch controlling the operon is turned on. The mRNA transcribed, the enzymes protein are translated, and tryptophan is synthesized. The tryptophan operon, the 5 genes that are needed to synthesize tryptophan in E. coli, are located next to each other in the operon. When tryptophan is plentiful, 2 tryptophan molecules bend to the pressure protein at the operator sequence. The physically blocks the RNA polymerase from transcribing the tryptophan is absent. The repressor protein does not bend to the operator and the genes are transcribed. So the operon includes 3 important regions: the coding regions, the operator, and the promoter. The coding region includes the genes for the 5 tryptophan biosynthesis enzymes. Just before the coding region is the transcriptional start site. So the promoter sequence to which RNA polymerase bends to initiate transcription is before or upstream of the transcriptional start site. So between the promoter and the transcriptional start site is the operator region. So operon is a fancy way of regulating genes, and it is usually made up of few genes that involve enzymes. Remember that enzymes are proteins with the ability to break down or build up substance that they act on. So RNA polymerase is a builder enzyme. RNA polymerase is needed in order to start transcription. Remember that transcription and translation are steps in protein synthesis. So protein synthesis, which means to make proteins, enzymes in this case. So RNA polymerase needs a promoter. A promoter is a sequence of DNA where RNA polymerase can bend to. So you will think that's get RNA polymerase attached to a promoter, and you will make your mRNA, which eventually will be used to make a protein. But there's another way sequence of DNA called an operator. The operator is a part of the DNA where something called a repressor can bend. A repressor, if bound to the operator, blocks RNA, so the RNA polymerase cannot move forward and no mRNA made, therefore no proteins.